Hello everyone, now welcome to Module 2 of Genome Annotation. So previously we did Module 1, which is the basic information module, and now we are going to do Module 2, which is sequence-based similarity data. So scroll down and you'll find Module 2. So open up your module. And the module, of course, is sequence-based similarity data. If you want to see instructions for the module, you just click here and that will take you to the instructions. The first thing we are going to do is a blast search. A blast search identifies proteins that are similar to the protein that you are working with. So what you do is you copy and paste, or you can actually control click, and this will open up a new tab, and this is the blast tab. So if you scroll up and down, you see this is NCBI blast, and just scroll down a little bit and click on protein blast, and that should take you to this page here, which is the protein blast page. Now what you need to do is go back to your notebook. So you'll see a whole bunch of tabs open up top. Don't close the tabs. Go back to your notebook and look at your protein sequence. Okay, so what you do is you copy and paste your protein sequence into the blast page. I've already done that. So now what BLAST is going to do, it's going to pull up proteins which are similar to your protein. Why do you want to find out this information? Because similar proteins will have a similar function. So what you want to do with genome annotation is figure out what function your gene is involved in. So what you do is you basically copy paste your protein sequence into BLAST. Okay, scroll down, make sure it's BLAST P, protein BLAST. Okay, choose the NR database, which is the default database. Scroll down a little bit more and then hit blast. Now this can take a little time. Let's see if we can do it quickly. Okay. So it's updating. So what we can do in the meantime is to see which domains are conserved. We'll let that go for a little while and click on this yellow tab right here. And what that takes us to is the conserved domains page. Okay, this is the conserved domains page. So if you go back to your notebook after doing the blast search, they will ask you to find the conserved domain. So while the blast search is going on, we might as well look at the conserved domains. So CDD is conserved domains, obviously, and what you need to put in your notebook is the cog number and the cog name. So let's go back to the conserved domains, and the cog number is this, COG, and what is it? It's a cobalamin biosynthesis protein. So already we're getting an idea of what this gene does, because it contains these conserved domains most of which are involved in cobalamin biosynthesis. So you go back to your notebook and you put in your cog number and your cog name. Okay, now let's go back to our BLAST search. Scroll down a little bit. Okay, so what they ask you from your BLAST search is the gene product name. So take the second hit because the first hit will probably be your own protein. So we had BLAST open here. Let's see if BLAST has completed. Yes, the BLAST search has completed. And here what you see is the conserved domain. So we can see that the protein belongs to the COBN-like superfamily here because it has conserved sequences which are responsible for cobalamin biosynthesis. So now what you'll see is the BLAST graphic, and in red is where this is probably your protein. The second one is where the second protein aligns with your protein. Third one, the alignment of the third protein, which was pulled up, and how well it aligns with your protein. If you scroll down, what you see here is your protein. This was our protein, cobalamin biosynthesis protein, Methanothermobacter thermoautotrophicus. So, what the BLAST search pulls up is similar proteins either in the same organism or in different organisms. So now you take the second hit. So the second hit is actually a cobalt chelatase, which is found in a different strain of Methanothermobacter thermoautotrophicus. But if you look all the way down, this gene or the protein it produces seems to chelate magnesium and may also be involved in cobalamin biosynthesis. Okay. Now go back to GenieAct, and what do they ask you for? They ask you for the gene product name, the top hit. Basically, we're taking the second hit because the top hit will be your protein. So what we found, the top hit was the cobalachelatase in Methanothermobacter thermoautotrophicus strain CAT2. Then it asks for the organism in which you found this hit. So you put in Methanothermobacter thermoautotrophicus, CAT2. It asks you for how well this protein aligned with your protein. So go back, and what we can see is we have to look at the alignment. So you scroll down and you look at the second alignment, Methanothermobacter thermoautotrophicus, and look at the gaps. So if you look at the gaps, it says 0. What this means is that this protein perfectly aligned with your protein and there are no gaps. So that's what you put in your lab notebook page. If you scroll down to the next protein, it will ask you for information on the next protein as well, which happens to be a magnesium chelatase from Methanothermobacter thermoautotrophicus, the other strain of this bacteria, or archaea actually. In the gaps column, it says 84, which means your protein aligned with this protein, but there were 84 gaps. So here you can see the gaps here where there was misalignment or no alignment. So this was your query sequence, which is your protein, and this was the protein we're looking at right now. And where you see these little dashes, that means there was no alignment, hence they are gaps. So put this information into your lab notebook. So let's go back to the lab notebook. Okay. Now, so there were no gaps, therefore the alignment length was the whole protein. So if you go back, it is to the first one. It is 1649. And then what else it asks for is the bit score. So you go in and you put in your bit score from the BLAST. So there's this table up top. And it has lots of different scores. It has the max score, total score, query cover, so on and so forth. And from here, you need to put in the bit score. Okay, so you have to look in the list table. So let's go back and look in the list table for the bit score. Here you go. This is your bit score. So then you put your bit score in your lab notebook, and then you basically copy paste the entire alignment of both sequences into your lab notebook. So basically what you're going to do is you can copy paste this entire block into your lab notebook. Then you do exactly the same thing for your next hit. Okay, so you do exactly the same thing for your next hit. Now you go back to your GeniAct notebook, and what we've done so far is looked at sequence-based similarity data in this module. We've pulled up a couple of proteins which are very similar in sequence to our protein of interest, and we've also seen that they have some role in chelating cobalt. Okay. So here what you did was you put the name of the gene, the name of the organism in which your similar protein was found, the alignment length, and the bit score. Okay. Then you put in the alignment of the query sequence and the second hit, because the top hit was your own protein. And you did the same for the second hit. Then what we did was we went to the conserved domains tab, and from the conserved domains tab we put in our cog number and our CD number and also the cog name. Okay. Now the next thing we do is we go to tea coffee to align the top 10 sequences. So here are the top 10 sequences. How did I get those top 10 sequences? You go to BLAST, go to your BLAST search again, go to your table, then you click off the top 11. Right? So the first one is your sequence and the top, and then the next are the top 10 sequences. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Then you scroll down. And you download these sequences onto your desktop, right? So now you have 11 sequences on your desktop, you copy and paste these sequences into your lab notebook. So let me just show you. There. So these are the sequences which I copied and pasted into my lab notebook. These are not aligned sequences, they are just raw sequences. Now what we're going to do is we are going to take these sequences and put them in tcopy to align the sequences. Why are we going to do this? Because we want the aligned sequences so we can plug the aligned sequences into WebLogo to generate a sequence logo. Okay, let's go back. So what you need to do is you need to go to tcopy. So what I'm going to do is I'm going to copy and paste this. Edit, copy, the new tab, edit, paste, and that should take me to the tcopy server. Now what tcopy will do, it will align these 11 sequences. So you go back to your GeniAct page, and all you do is copy and paste these 11 sequences into tcopy. Okay, make sure you get all of it. Okay. Copy, go back to tcopy, edit, paste. Okay. What happened? Edit, paste. Okay, now it's all pasted. You can also upload the file from the desktop because you've already saved the file. You can be notified by email if that's what you want. Then you hit submit. So while it's working on that, let's go back to our lab notebook. So what it will actually give back to you is the multiple sequence alignment. So once it gives you the multiple sequence alignment, you copy and paste it into your lab notebook. Okay, that was quick. So now this is the multiple sequence alignment. All you need to do, you can actually look at the cool colors. Here you go. Well, that's going to take a while. There you go. We have cool colors. And as you can see, similar amino acids will be in similar colors. Amino acids with the similar properties will be in similar colors. So you can now copy the entire thing and paste it back into your lab notebook. Once you've pasted it back into your lab notebook, the colors will actually go away. So, unfortunately. Always remember to save your work. So there's a save button at the bottom. There isn't a save button on mine because mine's already saved. So just save the entire thing. And this is the alignment of the top 10 sequences with your protein. So these are the top 10 proteins which are very, very similar to your protein. And this will help us determine the function of your protein. So your task is to determine the function of your protein because that is basically the function of the gene that you're trying to annotate or find a name for. So let's go back down here. So now you have your multiple sequence alignment. What we're going to do with the multiple sequence alignment is to plug it into WebLogo. So what you need to do is, I'm going to shut off some of my tabs which are open up top, is you need to go to web logo. So I'm going to copy and paste this into a new tab. And this is web logo. You need to go to web logo 3, okay, not to version 2. Click on web logo 3 and create your own logo. Now what you have to do is copy and paste the multiple sequence alignment into web logo. Okay, so we have the multiple sequence alignment. Just copy the whole thing into web logo. Okay, and then what you do is create logo, and that should create your web logo. So it's working. I've already created my logo. Then you download your logo onto the desktop and save it, and then you can copy and paste it into your annotation. Also, while you're doing the annotation, sometimes things tend to disappear, so always keep a copy in a Word file. So copy paste all the information that you're doing in your annotation into a Word file, so just in case you forget to save it or if there's a glitch in the system, you still have your Word file as a backup. So here is my web logo. What does the web logo tell me? The bigger the letter, the more the conservation, okay? Which means particular amino acids are conserved between these sequences. So here you can see the tryptophan at this position. So the numbers are the amino acid positions. Sometimes you might not be able to save your entire logo in your notebook, in which case just save a portion of it, but always save it on the desktop because you will need this to complete your genome annotation. So here we can see there aren't really many conserved sequences per se here. So if you see a region where there is a lot of large letters, then that region is conserved among the proteins. And why is that important? Because it might help us find out the function of that particular protein. So with that, we have completed module 2, which was the sequence-based similarity data, and we've found proteins that are similar in sequence to our protein of interest, the protein the gene of which we are annotating, and we've kind of sort of figured out that the function of this protein, and we found conserved domains in this protein. So what did we do in module 2? What we did was we did a blast search, and we copied and pasted data about the top 2 hits into blast search. Then we found the conserved domain designation and name. Then we used tCoffee to align the top 10 sequences. This is the alignment. And then we plugged the multiple sequence alignment into web logo 3. Very important, use web logo 3. And then what we did was we copied and pasted the web logo into our annotation notebook. The importance of the web logo is that it lets you see regions that are conserved among these proteins. And conserved regions usually have a similar function within these 10 proteins. Well, 11 if you count your own protein. And with that, you can, and then at the end you basically write in your comments and observations. So if there is a sequence which is highly conserved, you write in which sequences are highly conserved. Then you save your work, and then we'll go to the cellular location module, the next module, module 3, in which we will try and figure out in which cellular compartment this protein is located. So where in the cell this protein is located.